Sahana Rangal, Claire Jeffrey and Mohammed Johnson from the Department of Medicine, and also importantly, Cheryl Valentine and Karen Fenton from Project ECHO and Gastro Foundation. So we have a very interesting topic this week on the COVIDsortium, the healthcare worker study. This is going to be chaired by Graham Mankies. So over to you, Graham will introduce our two speakers for the day. Go ahead, Graham.
Graham, I'm not sure if you can hear us today. We can see Graham, we can't hear him.
So maybe whilst Graham comes online, I'll just start. So we're going to have two speakers today. There's James Moon. He's Professor of Cardiology at UCL London and leads the Cardiac MRI Advanced Heart Centre, Europe's largest cardiac centre. And he's going to be introducing the healthcare worker study, the one-year results. And then that's going to be followed by Ntobeko, Ntusi, who's going to look at the Cape Town group and look at their results with us. Yeah. As usual, we'll be starting with Mary Ann Davies, who's going to give us an epidemiology update in terms of how we're doing here locally in Cape Town, in the Western Cape. Mary Ann, over to you.
Thanks very much. Just waiting a second for the slides to come up. That's great, thanks. So we can go straight to the first slide, which is the one that is very familiar to everybody, which shows our first and second waves very clearly, and that we are in a lovely trough post second wave, and the arrow shows where we were on the 4th of March with really all our indicators having declined. But when we have everything dwarfed by the big waves, it's difficult to see small changes. So if we can go to the next slide. What we're going to be looking at is just the last month for the Western Cape province. Just waiting for the slide to appear. There's a little bit of a delay. There we go. Okay, in the middle at the top is the number of cases and the 7-day moving average. And a month ago, we were at about 180 cases a day. We've gone down to about 140 new cases a day in the province, which is about a 20% drop. We're going to look into that line a little bit more closely in the next slide, because if you look hard, you might think it's dipped and it's possibly gone up a bit. And the big question everybody asks me almost every day is, has the third wave started yet? So I'm going to talk to that a little bit in the next slide. Bottom left, you can see our proportion of positive tests has remained stable at about 5%. In the middle, number of new admissions per day has dropped by about 30%. And on the bottom right, the biggest decrease has been in our number of deaths per day, which has dropped by about 2/3 from 16 deaths down to less than 5 deaths a day in the last week or so. If we go to the next slide, I'm going to talk a little bit about looking for resurgence, which of course we're doing very, very closely at the moment. So this is just showing the Cape Town Metro because it illustrates what we're looking at very well. The dark blue line at the top is the number of new cases per day, the 7-day moving average in the number of new cases per day. And then the yellow line is the percentage change in that 7-day moving average. So essentially on a daily basis, it's the week-on-week change in the average cases in the last 7 days compared to essentially the 7 days before that. And the IMT, national IMT criteria for putting us into alert are if we go above a 10% increase in the 7-day moving average and stay there for 7 days, and responses if we go above 20% and stay there for 7 days. But what I want to illustrate is how public holidays wreak a little bit of havoc with those measures. So you can see that after we had a public holiday on the 23rd of March, we had a real dip in our 7-day moving average of cases, which measured the previous 7 days. And just after that, it looked like we were heading straight into alert and resurgence, caused all sorts of excitement and alarm. We didn't quite stay above 10% for 7 days, but as we got to the 30th of March, and we picked up all those tests that had been pushed from the 22nd of March into the next week, we went up to a 40% week-on-week increase in our 7-day moving average. And now, of course, with the public holidays over Easter, it's plummeted right down. So it's we watch it closely, but until we get back to some 5-day weeks, it's quite hard to actually interpret these results. And the bottom figure, which shows the number of tests per day, you can see the sort of gap-toothed appearance as we move towards the end of April with the public holidays, where we've got gaps, particularly in public sector testing, and our tests per week have actually dropped from about 24 to 22,000 per week. The one caveat to mention is that we're just looking at the moment to make sure that all the antigen tests are pulling through properly, because there may be, as facilities have changed to more antigen testing, we may be missing some test results there. So that's something we're looking at. We can go to the next slide. And this just shows that you see the same thing in the reproductive number when we have a whole lot of cases coming through following public holidays. And so we blipped above 1, but have now reassuringly gone back below 1, but continue to monitor closely. We go to the next slide. So what I'm going to spend most of the rest of my talk talking about is our latest Sentinel seroprevalence data, which is based, as I think everyone is familiar with, on using routine specimens for people attending healthcare for non-COVID reasons. And on the far right, you can see that during the middle 2 weeks of February, so very much post-wave 2, we really tried to do extensive seroprevalence testing in all the groups that we'd done previously. So people with HIV, diabetics, for the first time, including private sector patients and children. And I want to just acknowledge the people on the top left who were part of the big team effort that made it possible to test about over 5,000 specimens in a space of, or collect and test in a space of about 2 weeks. So we'll go to the results in the next slide. This is just to show the characteristics of patients that were included in that study. We tried very hard to get diabetic public sector representation across the whole province, so included from all districts and all sub-districts within the metro. Private sector diabetics only came from the metro, and people with HIV only from the metro, because that's who we tested previously. And children, it's very hard to get residual specimens, so we used Tygerberg and Red Cross as our source, and you can see at the bottom that about half of those were actually inpatients. A couple of things just to point out: the age distribution, which is important in terms of seroprevalence, was similar between our public and private sector diabetics, but as we expect, younger in people living with HIV, and of course in children. We had more women in our diabetic public sector compared to private, and also in our people with HIV. And then importantly, we hadn't really planned to include any inpatients, but some did slip into the sample, about 5% of the public sector diabetics and 10% of the private sector diabetics. And we were concerned that this might push up the seroprevalence in case these people were actually admitted with COVID. But in fact, in the public sector, the seroprevalence in the inpatients was actually lower than the outpatients. We don't know for the private sector because we haven't been able to link the data of inpatient status to the actual seroprevalence results, but that is one concern, and I'll talk to that a little bit later. We can go to the next slide. So firstly, just to look at the diabetics, which we think are probably the most representative of our adult population in general. And this is looking across the province. The pale orange results are from the end of wave 1 in October, November, and the dark orange are our more representative sample. You can see by the numbers from each district at the end of wave 2, you can see a big increase across all districts as we expect, but quite a bit of variation from 33% in West Coast to 47% in the metro. Go to the next slide. Here we're focusing in on the different groups in the metro in February. So on the left-hand side, again in orange, we have the diabetics as presented in the previous slide, and then the different groups. So much higher seroprevalence in the people with HIV, lower in children, and lowest of all in the private sector. And all of these are different from each other. And if we go to the next slide, I'm going to unpack this a little bit by sub-district and over time. So this is a busy slide, and just to orientate you, the blue results are all in people with HIV. The lightest shade is July of last year, moving through to February, the most recent results. The orange is the diabetics, and the grey is the private sector. And because we couldn't in advance select the sub-districts of the private sector, I've shown the numbers from each different sub-district, and you can see that it varies. And as we expect in the poorest sub-districts, we have the fewest private sector specimens, and in fact, we only got one specimen from Khayelitsha, so we haven't included that. I think a couple of things to point out. To start off with the diabetics, which again we think is our best representation of adults, at least in our public sector patients. Pretty much across every sub-district, the seroprevalence, the absolute increase was about 20% during the second wave, or at least 20%. So even Khayelitsha, which was sitting at 50% at the end of wave 1, still had a further 20% increase in the diabetic seroprevalence, right up to nearly 70%. The diabetics consistently had lower seroprevalence, with more variation across the sub-districts than the people living with HIV, and that's in keeping with people living with HIV generally being of poorer socioeconomic status. And the sub-districts where this was different was sub-districts that homogeneously are relatively poor, such as Khayelitsha and Klipfontein, where you can see the dark blue and the dark orange are most similar to each other. And then the private sector seroprevalence was consistently lower, in the order of 20 to 30%, except in Mitchell's Plain and Eastern. And in Mitchell's Plain, curiously, the private sector seroprevalence was actually slightly higher than the public sector. But those two sub-districts actually had slightly higher proportions of hospitalized patients, about 15%, and obviously the sample for Mitchell's Plain is small. So we're still busy trying to unpack that a bit further, and it may be that that number for Mitchell's Plain is really just too low to be reliable. If we go to the next slide, what I wanted to highlight here is, it's a slide that I think I've showed previously, is the seroprevalence at the end of wave 1 in Khayelitsha was sitting at about 50%. And if you look at the figure on the right, that seemed to offer some protection in terms of wave 2 deaths. So they had the lowest ratio of wave 2 to wave 1 deaths. If we look across all our metro sub-districts now, at least in the public sector, our diabetic seroprevalence is sitting at about 40 to 50%. We certainly hope this means there might be some protection against a subsequent wave, but I think we have to wait for the subsequent wave to really know what the answer will be. We can go to the next slide, which I think is looking at age and gender. This is looking at seroprevalence by age for the different groups, the private sector diabetics in black, otherwise diabetics in children in orange, and the people with HIV in blue. And just to show this pattern of highest seroprevalence in our young and working age adult populations, with a big drop-off by age, which supports prioritization of older people for phase 2 of vaccination. And interestingly, in our public sector patients, much higher seroprevalence in women compared to men, but we don't see this as much in the private sector, and it probably relates to the ability of women in our who use the public sector to socially distance, and perhaps related to economic activity and public transport use. And then finally, just to talk about, we use the Roche antibody test for all of these results, but you can see that in about 1,000 of the specimens, we also tested with Abbott. And I just want to highlight this in case you're reading papers or trying to interpret Abbott results. And what you can see is the top right is where they were both positive, but that there were a large number of tests in the bottom right, which were positive by Roche but negative by Abbott. And so our Roche positivity was about 46%, but Abbott only about 28%, which means if Roche is the gold standard, we have a sensitivity with Abbott of only about 60%. So really indicating that the Abbott results, and if you're reading any papers, are likely a substantial underestimate of true seroprevalence. And then lastly, I just thought since we're talking about healthcare workers, I would highlight that we have had 8,900 healthcare worker infections in the Western Cape, sadly 124 deaths, and you can see that clearly nurses by far the most affected category with nearly 4,000 nurses infected, but also to note that if you compare the second and first wave in health workers compared to the general population, the swamping of the first wave by the size of the second wave is actually relatively a little bit less in the health workers. And I'm sure the health worker studies might give us some clues about that. And then lastly, to talk about vaccines. If we go to the next slide. So you can see that, sorry, I can't quite see it on my picture, but you can see we've vaccinated in excess of about 50,000 health workers, and we expect to vaccinate about 67% of all our health workers with the Sisonke program before we move to what's going to be called phase 1B, which is when people like me will finally get vaccinated. Thanks very much.
Yeah, thanks very much, Marianne. Apologies, I wasn't able to introduce you. I was having technical problems, but thanks very much for that excellent update. You know, many aspects in terms of giving us an understanding of what's happening with COVID-19, and fascinating to see that some of our communities have up to 70% seroprevalence. And, you know, with time, we'll know what the impact of that is on a potential third wave. So thanks, Marianne. We're going to move on to our main session today, which is to hear from two speakers about healthcare worker cohort studies that have been done in tandem in London and in Cape Town. And these studies have really been a remarkably powerful mechanism to deepen our understanding of SARS-CoV-2 infection risks and dynamics, as well as the immune response and protection provided by previous infection and vaccination. The double act is going to be from James Moon and Ntobeko Ntusi. James and Ntobeko share in common that they've both either worked or studied in Cape Town and the UK. And both of them are cardiologists whose primary research field is cardiac MRI. But over the last year, they've refocused their group's research on COVID-19 in the form of these healthcare worker studies that have provided us, as I said, with remarkably important insights into SARS-CoV-2 infection and immunity. So James is going to start off with discussing the COVIDsortium healthcare worker study, one-year results. James is Professor of Cardiology at University College London and leads the cardiac MRI research unit at Bart's Heart Centre, Europe's largest cardiac centre. So thanks very much, James, for joining us this afternoon. We really look forward to hearing an update on your study.
Thank you, Graham. Can you hear me okay?
Yeah, I'm hearing you, and I can see
Fantastic.
the slides in full screen.
Great. Yes, so I'm representing a group of people that have come together over the year. And actually, I only realized today that I've not actually met most of these people, and we haven't had a chance to actually be in a room together. And yet, Danny, Rosemary, we talk every other day constantly on calls in this fantastic project that was set up thanks to the collaboration of many people. So to introduce it, just move my slides on. We were watching Bergamo and Italy in March, wondering what was going to happen. And we knew as cardiologists doing cardiac MRI that our research would not survive this. And we wondered how to reposition ourselves. And we had lots of possible scenarios that we could study. But thinking about the exponential function and that's what Ntobeko and I do every day with the power of our magnetic fields, we knew that you can't win a fight against it, and that societal resources in immunology and virology and intensive care would be consumed, and that if there was any research to do, it would have to be fast with atypical leadership and done as quickly as possible, deeply resourced and collaborative. And we published the modeling, and you can see the spike of the London first wave there. So what we did, working with my old friend Maddie Nursadehi, who we did MRCP with, we did that together at the Brompton in the past. We decided to bottle and capture mild disease and the earliest changes in what we thought would be 95% of infections who didn't end up in hospital. And so we did that fast. We basically went from idea to first recruit in 7 days and completion of recruiting 400 subjects in around about 2 weeks. And having made those documents and got all those permissions and patient information sheets, being academics, we thought that if this was a pandemic, we would, other people with capability of delivering a project like this may not have the capability to set it up. So we made those documents available to colleagues, and Ntobeko, having been a colleague in MRI, was one of those, and he then took this on and did a sister project in Cape Town. We were interested at that stage in the possibilities of differential vitamin D or the reverse seasonality of coronaviruses and ethnicity. Of course, we now know there's emergent strains as well, but we didn't think of that at the time. So we were taking samples basically, and healthcare workers are important in their own right, but they're also workforce representative, and they are captured in hospital and can come to work during a lockdown. So we tried to do a high dimensional sampling with a questionnaire that evolved over time as we went from worrying about travel or ibuprofen through to vitamins and household sizes over time. And this we delivered over first 16 weeks of weekly sampling, then 6 months, 3 months, 6 months, and then now 1 year, which we're doing today. It had a number of amendments, particularly putting in nested case control studies with large volumes of PBMCs, and we've got several of the groups on the line today about that. It was a difficult time. There was a lot of logistical issues. We ended up making our own swabs, for example, but we never missed a collection on a patient because of a lack of resources, which is quite remarkable, really. We had to reconfigure the CL3 lab, and Joe, Corinna, and Anya are on the call today. They did that over a weekend, and Marla set up large throughput PBMC processing along with 2 other labs to capture these samples. When we were doing well, we expanded the cohort to a scale that could use genetics to understand things. So we merged with Royal Free to get to 731, then 1,300 with Nottingham, and of course, Cape Town. We'll hear about 400 subjects in Cape Town. So what were the results? Well, we first published the design of the study, and that shows the number of people who were involved in it. I've put a QR code on each page if you wish to take a screenshot. The first result was of rates of infection in our healthcare workers, and these were people who were healthy enough to come to work and had a PCR swab, and we couldn't analyze them at the time, but you can see here that we were 7% active PCR positivity on the day of UK lockdown, and that the healthcare worker infection rates fell with community infection rates and not with hospital infection rates. Now, I'll show you later that we were fairly certain there's nosocomial transmission there, but by 6 weeks after lockdown, that none of the hospital staff were effectively infected. So even though our patients abandoned us, they shouldn't have been afraid at that stage of catching SARS-CoV-2 off our healthcare workers, and that was important. That was published in Lancet, and Tom Tribal was the first author. You can see what happened with seroconversion as well. If you look here, there are 2 cohorts, the first cohort we did and then the second one. And you can see that we captured the cohort before seroconversion. And in quite a number of patients, subjects, we have before, during, and after samples of their infection, which is quite important. And we have some very precious PBMC samples at the beginning, pre-infection, for comparison with larger aliquots afterwards. You can see also that there's a skew in the infection rates because we think that in the first cohort, there are a number of people isolating at home who were not included. And then in the second cohort, they came in. So there's a different seroprevalence rate. This is with Roche anti-MP as well. When we look at who got infection, and this is not severe disease because none of our patients, really, subjects got severe disease. They are some of the things you'd expect. So household exposure was a risk. Being Black in London and Nottingham, but not any other Asian or mixed ethnicity has a ratio of 2. And then the other thing that was interesting was that our ICU workers had half the prevalence, suggesting that the PPE availability or perhaps effective use of it in ITU was protective. But there were no other associations with age, sex, postcode or role that we found. Reflecting on Marianne's data, we looked at the antibody kinetics, and this is work by Benny Chain with Tim Brooks at Porton Down and UCL with first author Charlotte Manesty. You can see here the results of the positives in 157 subjects across 12,990 samples. And you can see the time course of the Euroimmune anti-S and Roche anti-MP results. And that was modeled by Benny, and you can see that anti-S here had differential kinetics, and this was probably the Euroimmune assay partly as well as the biology to Roche, and we conclude a bit like probably the Abbott data that was hinted at earlier, that anti-S would be bad for seroprevalence studies. Within the cohort, we then did the first nested study, and we matched 157 positives or 74 positives with negatives. And looked at the T and B cell responses, or especially the T cells here. And this is Danny Altman, who's on the call with Rosemary, Marla, and Anya, who did the neutralization. And I can't really give justice to these data, but what you can see here is the 2 columns of results. One is ordered at the bottom by the neutralization response at 16 weeks of our healthcare workers, some with a very high response and just about 8 with no response. And then you can see above exactly the same patient's T cell response to different epitopes or pools of epitopes. And the other column on the right is the same data, but this time ordered by the T cell response. And you can see that there's discordance, but the vast majority of subjects have one or other responses, but not everyone has both at the same time. Very important data, and I think this will become a foundation for more understanding of the heterogeneity of the human response over time. If you do have questions, Rosemary and Danny and also Anya are actually on the call afterwards to query these data. Now, we also looked at the heart a little bit later in convalescence, and we saw that if you were troponin positive and been in hospital, so different cohort, there was a lot of scarring and damage in the heart with some residual inflammation. We never, in a single time point, can unpick which is cause and effect, but we highlighted just how much cardiac injury was sustained by those with survivors of severe disease. What about our healthcare workers who had almost entirely mild disease? Well, when we looked at 6 months using best available technology, meticulously done, this isn't a sort of first wave study. This is as good as we could do at any time. With contemporary controls, we found no detectable cardiovascular difference between healthcare workers who'd seroconverted and healthcare workers who had not. And this really is the best technology. So by 6 months, there were no sequelae. Lots of our healthcare workers had abnormalities, and you would expect that when you measure at least 10 parameters and define abnormality by 2 standard deviations, but they weren't co-segregating with prior COVID, which is a very reassuring result and slightly discordant with some of the results in the literature. So we were moving through at this stage to a sort of post-vaccine stage, and we looked, thanks to Rosemary, but also with Ash, who's on the line, and Charlotte leading, at the response to vaccination. And this is the Pfizer vaccination, and we took blood samples in 51 subjects at a median of 22 days post first dose. And what we saw is, if you look at the graph here, that if you, this is the uninfected got a got about 100 on the Roche anti-S, it's units, not international units response, which was the same as their peak response in the infected. So this is actually peak response in the infected. But when you gave that priming dose of Pfizer mRNA vaccine, it put another 140-fold times higher antibody response on that. So that single dose primer actually acted as a boost, which I think people are now familiar with. But we were lucky enough and made our own luck, thanks to incredible work in the team, to get that published in Lancet first. And it suggested a strategy for how to make vaccine go further, and may also translate into strategies for people who, if we ever need boosters, for example, in the future. Maddie looked at the transcriptomics, and this is exciting data using those PAX gene tubes because samples were taken before, during, and after infection. And here you can see results that's in preprint at the moment, looking at known transcriptomic responses to respiratory infections. And what you see here is type 1 interferon response components going up and down in a wave, but you see those host responses a week before the PCR is positive. Suggesting that host innate immune diagnostic tests for COVID, or at least here viral infection, could form part of acute care testing going forwards. This was using known signatures. If you look at the whole blood transcriptome and have a more generalized approach, what you can see is the same thing of a response before the PCR goes positive, here with again an interferon response occurring early, and then you see a cell proliferation response, which seems to be TCR clonal expansion, CD8 greater than CD4. And from your wonderful libraries you have, these seem to be COVID specific or databases, and that is a unique feature of COVID-19 compared with other viral responses. And the two sides, the interferon type 1 interferon response and the cell proliferation, seem to be independent. So this is a signature of sort of healing innate and then adaptive immunity, because none of these subjects became hospitalized. So that's the published studies. We have a number of other studies going through with a number of PIs, as you can see here. We're cardiologists. We were doing lots of CMR research, and we repurposed into trying to capture mild disease, as it turned out, in healthcare workers as a bioresource for other people to use. And I'm just very grateful to all the immunologists who have used these samples and put together some of the puzzle, and of course for Ntobeko for doing a sister study in Cape Town, which I'm looking forward to hearing about. This is the team, or the leaders of the team. Of course, there are many, many other people as well. Thank you.
Thanks very much, James, for that excellent overview and a really highly productive study in terms of the range of outputs that you've had to date. And I'm sure we're going to see a lot more from the specimens and data that you've collected, informing our understanding of the disease. So I'm not going to take questions at this time. We'll move on to Ntobeko's presentation and then have a question and discussion session afterwards. So the next speaker is going to be Ntobeko Ntusi, and Ntobeko is the head and chair of medicine at the University of Cape Town and Groote Hospital. And Ntobeko's been leading the COVIDsortium study in Cape Town and is going to present on that. So over to you, Ntobeko.
Ntobeko, just unmute, please.
Are you able to hear me?
Can hear you now, Ntobeko.
Oh, wonderful. I'm just going to upload the slides again. Sorry, my unmute there froze. Can you see my slides?
Not yet.
And now?
No.
Oh. Maybe Mark and Wendy, in the interest of time, are you able to show them from your side?
We
Because
certainly can.
let's
Just
do
try
that.
one. Just try one more time. I've just reset the system. But if not, we will just load up. I'm going to load up for you. All right.
Okay.
Okay. Just ask me to advance, please.
Wonderful. If you can advance to the next slide. So these are my disclosures, none of which have any relevance for today's talk, except to say that I feel a little bit like a wolf in a sheep's skin, being a cardiologist, talking to you today mostly about immunology. Next slide, please. So James has already given much of the background to this project, and really from the beginning, the idea of this COVID consortium was to develop a customized disease-specific understanding of COVID-19 and to use this bioresource to answer a number of critically important questions. Next slide, please. So from the beginning, we had quite a number of hypotheses that we wanted to test, and I'm not going to go through these in detail, but really today I'll be focusing on showing some of the work we've done looking at immunological phenotypes, as well as looking at the question of immune durability. And we have other streams of work ongoing or planned to look at the other aspects of these hypotheses. Next slide, please, Mark. So the design of the study, we looked at 400 healthcare workers who at baseline had a questionnaire and sampling with bloods and nasal swabs. And the original plan was to follow them up 2 weekly, but we ended up going 4 weekly for logistical reasons. And at 6 months, we then swapped to 6 monthly follow-up, which we plan to continue for the next 2 years. Next slide, please. So who makes up our cohort? Well, they are a young, relatively healthy cohort of healthcare workers, mostly doctors and nurses, with a mean age of about 40. You can see that 75% female and 25% male, and relatively healthy, the vast majority, almost 350, without any comorbidities, and the cardiovascular comorbidities, including predominantly hypertension, diabetes, and obesity. Next slide, please. And this really shows how the study has been progressing thus far, and the different measures at different time points and some of the demographic details described. Next slide, please. And so the first result slide really shows the high number of infections in this cohort. It's a really wonderfully curated cohort. We are able to have very comprehensive phenotypes, as well as the exact time points when people were infected, and so can study the dynamics of immunity over time. And you can see that about a third of patients were confirmed to be RT-PCR positive, and 73 of these infected during the first wave, and another 42 infected during the second wave. Next slide, please. And when using a validated in-house RBD ELISA, 131 out of 365 In other words, 36% were seronegative, and this was up to 8 months after infection. However, in total, on the antibody studies, an additional 17% of healthcare workers were seropositive, therefore bringing the cohort to 47% who were either PCR positive or seropositive. And I think it's important to highlight that this nearly 50% positivity rate may in fact be an underestimate, because the antibody ELISA test we are using is only to the RBD and not to the spike protein. We have conducted testing in Jonathan Blackburn's lab with the antibody test that looks at both of these, but I don't have those results to show. Next slide, please. And in a recent publication 2 days ago in the New England, together with Penny Moore's group, we showed that most of the infections from October onwards in Cape Town were due to the 501Y variant. And so looking at the infections at different time points, using October as a cutoff, we can then safely categorize infections into those due to the wild type or original strain, and those that we think are due to the variant. And of those, we've selected 53 from the first wave and 21 from the second wave, where we have performed detailed immunological analysis. And I'll show the results in the next slide. Thank you, Mark. So looking at the cohort that has been selected for this detailed immunological analysis, you'll see that they are relatively young, predominantly female, and relatively healthy, most of them asymptomatic or mildly asymptomatic, and with very few comorbidities. And in terms of their clinical roles, there was a wide range, and they also worked in different areas within the hospital. Next slide, please. And we have, in collaboration with Georgia Schaefer, performed a range of immunological investigations to characterize immune memory after infection in those who were PCR positive. And the neutralizing antibodies were tested using a pseudovirus neutralizing assay against the original strain from Penny Moore's lab. And what you can see is that we have people who are tested at an interval of more than 4 months. And the key take-home message from this slide is that the responses to these neutralizing antibodies are maintained over time. And the neutralization titer remains stable over time. Of interest for us is if you go back to the previous slide, please. If you look at this figure, I'm not sure if you can see my mouse, probably not. There's about 6 or 7 individuals where there is a more than 10, sometimes more than 100-fold increase in the titer, which we think is likely to represent reinfections, but we need to characterize this cohort in greater detail. Next slide, please. And when we look at the PBMCs that were stimulated with peptide pools that span both the nuclear capsid and the spike proteins of SARS-CoV-2, one can see a classical T helper 1 response with cytokines TNF alpha, interleukin 2, and interferon gamma expressed at higher levels. We also saw clear CD4 positive and CD8 positive cell responses. Next slide, please. And when we look at the phenotypes of T cell responses, one can see that the CD4 responses, as has been published by other groups, were much higher in SARS-CoV-2 infections compared to those with CD8 responses. In our cohort, 91% showing CD4 positivity, as opposed to 62% showing CD8 positivity. Importantly, in the graph on the right, we also see a higher magnitude of CD4 positive T cells. Next slide. So on flow cytometry, we also looked at the cytokines associated either to CD4 or CD8 positive cells. And one can see a distinct functional profile in the figure on the left with IL-2 being the predominant cytokine in those who were CD4 positive, as opposed to interferon gamma being the dominant cytokine in those with CD8 responses. Next slide, please. And confirming what we saw in the previous slide, one can also appreciate polyfunctional profiles of T cells. And so the colors in the two pie charts explained in the scale at the bottom. So for instance, red refers to those who are positive for all three of these cytokines. And you can see a differential expression of these cytokines in those with CD4 and CD8 T cell responses. Next slide, please. And if we look at the contribution of spike and nucleocapsid to T cell responses, in those who were CD4 positive, we can see that S and N were co-dominant antigens, as opposed to those who were CD8 positive, where we saw a much higher magnitude of responses to N rather than S proteins. Next slide, please. And looking at the temporal trends of CD8 and CD4 responses, we can see that these are sustained over time. In other words, we show during the course of follow-up of these patients over 7 months, durability of the immune responses, which is very reassuring. Next slide, please. And when we look at T cell responses, comparing the CD4 and CD8 T cell responses in the first and second wave, first wave notated in blue and the second wave infections in red, probably reflecting recency of infection. There is a higher median titer in the second wave, but this was not statistically different. So we see that the T cell responses between the original variant and the 501 YV2 variant are not different. Next slide, please. And a question that a lot of people are interested in is what is the rate of reinfection in this cohort? We have out of 400 two people where we have definite evidence of reinfection. As I've showed you, there's about 7 individuals who had much higher titers after 4 months of follow-up, but it needs to be confirmed on RT-PCR testing as well as sequencing to confirm that these were indeed repeat infections. But the take home for us so far is really that the risk of reinfection appears to be low. Next slide. So to summarize, I've shown you that in this cohort of healthcare workers who are relatively young and healthy with minimal comorbidity and mild COVID-19 disease, there was a high number of asymptomatic infections. And importantly, convalescent healthcare workers who experienced mild disease mount durable neutralizing antibody and T-cell memory responses to SARS-CoV-2 up to 7 months after infection. The rates of reinfection appear to be low in our study, and we have not presented, in the interest of time, the other streams of work that are being done in this cohort. Next slide. I thought I would end off just by indicating some of the areas of interest for us, and one of the main ones is with all of these having been enrolled as part of the Sisonke phase 3B study and recently vaccinated, is to look at the effect of prior infection on vaccine responses, and so to look at immunological phenotypes in those infected in the first wave, in those infected in the second wave, as well as in those without prior infection. Next slide, please. And this work will include a detailed characterization of the T-cell and antibody responses with planned work to look at both pseudovirus and live virus neutralization in collaboration with Penny Moore and Alex Sigal, and of course Wendy Beggars being the one driving the immunological work in Cape Town. Next slide. So I wish to end by acknowledging a few people, and Wendy Beggars has really been the brains behind all the immunology, who's taught me a great deal about generosity, collaboration, and has been a lot of fun to work with in the last few months, as well as members of her group who've led this work. We also have several other investigators at UCT, and I wish to acknowledge members of our department and colleagues and staff in the hospital who have been happy to participate as participants in this cohort. Our collaborators are in the UK, as well as Penny Moore, Alex, and Georgia Schaefer. And I must really thank members of my own research group who have adapted very well in shifting the focus from the work that we do traditionally, and in particular, Lionel Chinoi, Matilda Menon, Sango Skelem, Olukayede Aremu, and Daniel Mutitu, who have led much of the work that I've presented today. Thank you very much.
Great. Thanks so much, Ntobeko, for another really outstanding talk, highlighting, a study that has and is going to, I'm sure, continue to contribute important understanding on the biology of COVID-19 and the immunity to it. So, and really congratulations to both you and James for the foresight and initiative and rapidity with which you got these cohort studies going, and transforming your research groups from cardiology research groups into infection and immunity research groups that and contributing to knowledge in this way. So thanks very much. We're going to move on to the question and discussion time, and maybe I can just start off with a question to both James and Ntobeko, to just kind of take a step back and say, you know, what is the, I think you presented the cumulative incidence in the two cohorts, and there are some differences, and, you know, looking at the infection profiles in the London cohort and the Cape Town cohort, are there differences that you see in terms of who's been infected, who's been protected? What are the risk groups in terms of the places that healthcare workers were working, or the profile of the work that they were doing? Just any initial thoughts on that?
James?
Thank you, Ntobeko. Thank you, Graham. So we wanted to look for that, and in fact, originally we were going to track people through the hospital and all those sorts of things, but we have quite crude data, to be honest, and the only role signature we found was that ICU staff were protected. We found that, but it was found better by other national consortia who showed that that protection was not present in early March, but kicked in by late March. And by April, none of the UK ICU staff got infected. And I think that shows that, firstly, that there is nosocomial transmission when uncontrolled in a hospital. And secondly, that it is possible to have completely effective PPE and social distancing strategies. Age, we were only, because all of our subjects were mild, we could only look at infection versus no infection. And we found no sex or socioeconomic aspects of that. Healthcare workers are all employed on the same salary, so they control for some aspects, except for one ethnicity, which was Black in London. The presence of an infection in a household, and I think, and Black people had twice the rate in London and not in. I don't know if that is a geographical issue or not.
Thanks, Graham. So in our cohort, we thought that people would be recruited in a particular area of the hospital where they would work, and we could follow them up over time and see if the location of your work mattered. But of course, a lot of interns, a lot of registrars and medical officers constantly move around between different areas in the hospital, as well as different hospitals. So it hasn't been as easy to do that. Even amongst the nursing cohort, a lot of them were moved from OPD to ICU to medical wards to theater. And so people have not really stayed in a single area. We have looked and have not really seen a clear signal to suggest that there are differences by age, gender, or I mean, most of this cohort is relatively healthy. The comorbidities were relatively low, but there's been no significant signal for what drivers of infection may be.
Thanks, Ntobeko. James, I just want to, before opening it up to questions, and I encourage people to type questions in the chat function, and Mark Sandrup will curate those. But James, I just wanted to go back to the issue of ICU staff being relatively protected, and wonder if that's not, you know, obviously PPE practices and infection control practices could play a role, but it could be a function of the stage of disease at which the patients are being admitted to ICU, and, you know, that they've had they've had infection and disease for longer than those patients who are initially admitted to hospital. And so that by the time people usually require admission to ICU, they might have been symptomatic for more than the 5 days at which they're highly infectious. And so that it's possible that even though the patients have more severe disease, they've got less infectious disease by the time they reach ICU. I'm just interested in your thoughts on that.
So absolutely, I think that is completely true. They've been through a journey. There was one point in March where we couldn't even test our own patients for SARS-CoV-2. And we know that some of our, at least a couple of our infectious endocarditis patients had COVID-19 and were seen daily on medical ward rounds unsuspectingly. And similarly with some of the acute thrombotic infarcts, they were actually COVID precipitated. So the ICU was protected by potentially by intubated patients. They also have 12.5-hour shifts. They have their breaks at separate times. And they're just more knowing. We didn't fit test for about 4 months for our FFP3 masks. Absolutely, I agree that there's more than just the presence, the availability of PPE to that signal, I think.
Thanks. So I'm going to hand back to Mark, Sandra. Mark, are there questions coming through the chat?
Yeah, thanks, Graham. I've just reposted 2 so everyone can see them. Maybe we just start with the first one, perhaps just back at James. The question is the signal you spoke of around Black people living in London being more vulnerable. You know, did they perhaps have more comorbidities or live in households which more people were essential services? Given that potentially socioeconomic factors are not at play in that environment, any explanation for that, perhaps?
So we measured things, but we probably didn't measure everything. But we did correct for socioeconomic status by postcode, index of multiple deprivation, and we corrected for role in the hospital. And we looked at single parameters like household size and distance traveled. And there was no explanation in what we saw. So it's possible that in that first wave, we were looking at something slightly more biological and social, but socioeconomic, but I don't know. There's also a signal, for example, if we look at the black people in Nottingham and London, in the genotyping we did, that there were differences. So just to say Black doesn't seem like a very granular category. But at the time in the UK, there was this sort of group called BAME, Black Asian mixed ethnicity, that were all being lumped together. And so one of the things we were trying to say was, look, you can't just group people by this catch-all BAME phrase. But with 1,300 subjects, we weren't powerful enough to get further into that. I also think it was important to try and send a signal to differentiate between severe disease and probability of catching disease in the first place, which was something that wasn't being well described in sort of April. Like it was a real focus on who was getting very sick.
Thank you, James. Professor Ntusi, question for clarity. I did post it. What proportion of the healthcare workers were HIV positive? Was their CD response more blunted compared, their CD4 response more blunted compared to HIV negative participants?
12 of the 400 participants are HIV infected, we haven't looked at the T-cell responses by HIV status, so I'm not able to comment on that question.
Thank you. Graham, that's it at the moment.
Okay, I do have one further question, and if there are any more questions that come up, let us know, Mark. But just to both James and Ntobeko, just to focus in on the cardiac injury issue. And you know, I think, James, you mentioned the very reassuring findings in healthcare workers that there doesn't seem to be any residual injury at 6 months. And you know, as you said, that does contrast with some other studies which have suggested that, and I think it might be to the fact that you've got very appropriate controls in your study and able to answer that question in that way. But I'm just wondering whether your view on how common it is in people who've had more severe COVID, residual cardiac injury, and whether you can just share some of the work that you've done or planning to do, and work that's in the literature regarding residual cardiac damage, a few months out from a severe episode of COVID.
You want to go for it, James?
Okay, so so if you take, we looked at 3 hospitals patients who were troponin positive, and there was about a third rate of people who'd been ventilated. So this is a properly severe cohort, and we looked a month after discharge, or within a month of discharge. And there is a survivor bias here because the trop positivity was a predictor of death as well. And most of these subjects have normal ejection fraction in their hearts, but the prevalence of scar is really high. It is not just myocarditis. There is thrombotic looking infarcts. There is stuff that could be myocarditis, but could also be microangiopathic thrombosis sequelae, we don't know. But even seeing those high rates, we don't get a good handle on whether these pre-existing damage predisposed for severe COVID, or whether it was a consequence of the severe COVID, because we really don't have a baseline scan in a population, see if they get severe COVID and then scan again type study design. We thought about whether we could get together and do that, but it's very challenging. So I think there are lots of cause association versus effect questions still ongoing. And I think I do feel that there is some blurring of the difference between long COVID after mild community COVID and the sort of post-ITU sequelae that many of the subjects that we're going to be seeing in cardiology have sustained.
Thanks. Ntobeko, anything from your side?
I mean, I agree completely with James. The healthcare cohorts from the UK and from us are patients who are relatively healthy, and so it makes sense that they had mild infections. I'm not sure about your cohort, James, but in ours we didn't have a single hospitalization out of the 400 people recruited, and so one wouldn't expect to have extensive ongoing myocardial inflammation or evidence of myocardial injury in the absence of persisting symptoms. But in those who've been ventilated and treated in ICU with very high evidence of cardiac injury, there's been evidence even in the early days from China that those not only have increased mortality, but have increased evidence of cardiac injury. And a number of CMR studies have confirmed that. I think it's probably an area where there's a fair bit of noise, and there's currently about 50 or so CMR studies globally looking at these questions. And I think in the next 12 months, we'll have much greater clarity.
Okay, the signal will arise above the noise. Yeah, and then just perhaps one more question, James, you're welcome to bring in Ntobeko, any of your immunology colleagues to help address this. But the issue is those people in the cohort who did not become infected with SARS-CoV-2, and whether we can learn something about whether there were protective immune responses in those patients by looking at cross-reactive T cells to other coronaviruses. Is there any plan to look at that, and do you think you will be able to address questions like that from your cohort?
Graham, I've got Marla Maney, who is on the line, who is interested in this area, but she didn't know that she might be asked to talk. And I'm just giving time in case anybody wants to unmute her so she can answer. There she is.
Hi,
Marla,
hi everybody.
Would you be able to address that?
Yeah, I mean, we're looking at that at the moment. And in fact, I was about to ask you if in your immunology studies you've looked at all at the uninfected, well, the group who was seronegative, should we say? Because we are seeing some interesting T-cell responses in those who remain seronegative with the same level of exposure and same demographics as the infected group. We're still trying to tie down whether those are cross-reactive to common cold coronaviruses, but it certainly looks like some of them may be.
And have you compared, the proportion that are positive among those who developed COVID versus those who remain uninfected?
Yeah, so interestingly, we do see an enrichment of certain responses in the group who remained seronegative to some regions of the virus. So I don't know if you've done T-cell responses to anything else other than spike and nuclear protein in any of your studies.
Ntobeko might be able to.
So I'd like to invite Wendy Burgers, if she's still on the call, to perhaps comment on both questions.
I'm just finding her. I think I have unmuted her. Yeah,
I can see Wendy there.
Hi, thanks for the opportunity. So that was in fact the endemic coronavirus immunity was one of the most interesting things we wanted to look at right at the beginning, but we haven't done it yet, simply because then, because we wanted to describe the SARS-CoV-2 responses first, but I think it would be still really interesting to look at that both at the antibody and T-cell level. We're starting to do that in some of the pediatric cohorts and collaborations that we're involved in, but could certainly extend those to this cohort. That would be really interesting.
Yeah, well, that would be great. It would be lovely to stay in touch about that. Do you have peptides to some of the other regions that you're looking at, as well as spike and nuclear protein?
We've only got matrix. In addition to that, we've only got matrix. So okay. Yeah.
Just that we think that the ORF1 region looks very interesting for cross-reactivity. So maybe we can try and see if we can do something together on that.
That'd be great. Thank you.
Graham, I've also got Danny Altman and Rosemary Boyton on the call as well on that T-cell side, and I'm just mentioning that because the question was so obviously for Marla, but they have also been looking at the T-cells, and it's my hope that Wendy and her group with Marla and Danny maybe have a further dialogue, especially as Ntobeko and myself are both cardiologists and actually would rather put an MRI scanner in any room where there's a bunch of freezers. So please, immunologists, use up the samples to answer important questions.
Okay, so I think that's a great way to wrap up, because it leads to new collaborative fields of investigation potentially, and a potential further outcomes from these two very important studies. So just to say thank you to Marianne and then James and Ntobeko for excellent presentations today, and to the attendees for the sustained interest and enthusiasm in participating in this COVID Echo webinar series. And we're going to continue with having monthly webinars. We'll send out the advert in around 3 weeks' time for the next COVID webinar. So back to you, Mark and Wendy.
Thank you, Graham, and we'll see you in a month's time. And thank you. We remain grateful to Project Echo, Gastro Foundation, and all the support from the Department of Medicine. Thank you very much. Be well.
Thank you. Bye.